change in genes needs insertion deletion. But now we have CRISPR, an acronym for clustered regularly interspaced short palindromic repeats. It is fast, cheap, precise, and relatively easily. It is a genome editing tool that is creating a buzz in the science world. It is faster, cheaper, and more accurate than previous techniques of editing DNA and has a wide range of potential applications.
That researchers create a piece of RNA with a guide sequence which is complementary to a targeted bit of DNA in the host's genome. In other words, if the DNA has a sequence that reads 5' GGCTAT 3', then the RNA guide sequence is exactly the opposite and reads 3' CCGAUA 5'. And remember, it's a U for uracil instead of a T for thymidine because the guide sequence is made of RNA and not DNA. The Cas9 protein then attaches to the RNA and the whole thing binds to the target DNA sequence in the host genome. The Cas9 RNA complex then makes a double-strand cut in the genomic DNA, and an alternative piece of DNA can be spliced in right at that spot. CRISPR-Cas9 technology works in a variety of cell types and organisms, and it's been used to study diseases and generate tissues from stem cells like heart muscle tissue and neuronal tissue. Now, it's also possible to treat a whole multicellular organism with genome editing. For example, a mouse with a liver disease due to a genetic defect was treated with a CRISPR-Cas9 mediated genetic change, and it improved the mouse's symptoms. One important point to note about this mouse example, however, is that the change was made to somatic cells rather than germline cells, meaning these genomic modifications aren't passed to the next generation. That said, CRISPR-Cas9 technology is able to alter the DNA in germline cells, and if that's done, then the engineered changes can be transmitted across generations. And this has been done in several organisms, including mice, monkeys, and most recently in humans. Last month, Chinese scientist He Jiankui claimed at a conference that he has edited the genes of twin girls using CRISPR-Cas9 technology. He specifically altered the CCR5 gene, making the carrier resistant to some strains of HIV. He made the change in 2 embryos, which were then implanted in a woman and carried to term. Reportedly, the unidentified twins were born and are healthy and at home with their parents. This announcement has led to an international moral outcry. First off, Jiankui acted in secrecy and didn't even tell his institution about his experiments. Second, it's not clear if there was proper informed consent from parents before the procedure was done. Third, the procedure didn't have a known medical need since neither of the infants had an HIV infection, and the procedure inactivated a healthy gene. Fourth, genome editing technology is still in its infancy, and there's no global consensus about using gene editing technology on human subjects. In addition, the risks of doing this to human embryos remains difficult to predict. For example, according to Jiankui's presentation, not every cell in the embryos was edited by CRISPR-Cas9. One possible outcome of this is that the twins may be heterozygous for the edited gene, meaning each cell has a normal and an edited copy of the CCR5 genes, in which case they may not even be resistant to HIV. Another outcome is that the twins may be mosaic, meaning that some of their cells have 2 copies of the normal gene, while the other cells have 2 copies of the edited gene, meaning that they'd only be resistant to HIV if the immune cells end up with the edited gene. In addition, the mutation made to the CCR5 gene didn't exactly match the known variation of the gene that confers HIV resistance. This means it's not clear if these untested changes will provide the same benefits, or if they'll introduce new unintended consequences. All this said, while the research appears to be rushed, and the fallout from it may have consequences that impact the scientific community for years, there is support for the idea that germline genome editing of human embryos could someday be used ethically to prevent devastating genetic diseases like cystic fibrosis or muscular dystrophy. The general consensus is that we're just not there yet, and that the scientists have moved quicker than the ethicists.
How does it work? System consists of two key molecules that introduce a change mutation into the DNA. These are an enzyme called Cas9. This acts as a pair of molecular scissors that can cut the two strands of DNA at a specific location in the genome so that bits of DNA can then be added or removed. A piece of RNA called guide RNA runner. This consists of a small piece of pre-designed RNA sequence about 20 bases long, located within a longer RNA scaffold. The scaffold part binds to DNA and the pre-designed sequence guides Cas9 to the right part of the genome. This makes sure that the Cas9 enzyme cuts at the right point in the genome. How was it developed? Some bacteria have a similar, built-in, gene editing system to the CRISPR-Cas9 system that they use to respond to invading pathogens. Like viruses, much like an immune system. Using CRISPR, the bacteria snip out parts of the virus DNA and keep a bit of it behind to help them recognize and defend against the virus next time it attacks. Scientists adapted this system so that it could be used in other cells from animals, including mice and humans. What's the future of CRISPR-Cas9? It is likely to be many years before CRISPR-Cas9 is used routinely in humans. Much research is still focusing on its use in animal models or isolated human cells, with the aim to eventually use the technology to routinely treat diseases in humans. There is a lot of work focusing on eliminating off-target effects, where the CRISPR-Cas9 system cuts at a different gene to the one that was intended to be edited. CRISPR-Cas9 looks like this.
Yeah.